The next speaker is David Piña. He's a member of the Soft Materials Technical Scientific Service from ICMAB. He holds a degree in nanoscience and nanotechnology and a master in chemical engineering. And he will talk about unraveling and quantifying molecular biointeractions through ITC and SPR.
Okay, thank you, Mark. Can you hear me right? Yes. Yes, so as Mark presented me, I work in the self-service here at ICMAB, and here we have a couple of techniques for characterizing molecular interaction. And so let's start this presentation on why, as from the biomedical point of view, why it might be interesting for you to characterize molecular interactions. Well, they are happening inside you. You cannot escape from molecular interactions. They are all the time happening. So, for example, in the treatment of COVID, some people were using antibodies to treat the COVID-19 infection. So that's a molecular interaction, of course. Also in the pandemic of opioids with all this topic of the fentanyl in America and so on. There's also a lot of scientists trying to understand or to comprehend how different molecules bind to these opioid receptors. Or simply in the cancer field. So we have a lot of protein-protein interactions that deal with blocking some kind of molecular pathways, biological pathways that promote or suppresses the presence of tumors. And in reality, in a lot of fields, we can find molecular interactions. As I was telling before, molecular interactions are everywhere. So again, from the point of view from biomedicine, it's very important to understand the disease that causes mechanisms or in order to treat them, in order to prove the therapeutics, because these diseases might be caused for interruption of a molecular interaction. Also, if you're developing some drugs in order to treat these diseases, or you are trying to diagnose or characterize, you need to understand how these small molecules that are drugs are going to interact with your biological receptors, or which biomarkers indicates which disease. And in general, let's say that in all biology, in order to map all the processes that happen inside the cells, so it's very interesting to quantify how this, what these molecular interactions mean, right? So in this seminar, since we're talking about today about techniques, I'm not going to talk about molecular interactions itself, but we're going to talk about two different techniques in order to quantify these molecular interactions. The first one will be isothermal titration calorimetry, or ITC, which is a solution-based detection that gives you the thermodynamics, or in simple words, why does these molecules bind. And also we're going to later to talk about surface plasma resonance, or SPR, which is a surface-based technique that gives us information about the kinetics, or how strong these molecules bind. Okay, so we're going to talk about how they work, and then a couple of samples for each, and then why one of them might suit you better than the other one, their advantages and limitations. Okay, so let's start with ITC. Okay, so this is a representation of how ITC works. We have a syringe, and then below we have an adiabatic chamber. If we zoom in here inside, what we have in the syringe, it's a titrant, so a molecule in solution that we're going to inject it inside this sample cell, which has another molecule that hopefully they bind to each other, right? And inside this adiabatic chamber, what we have in actually is two cells, the sample cell and the reference cells that is loaded with the same media or buffer that in the sample cell. And there is a potentiostat in this machine that tries to get all the time the same temperature into the sample cell and the reference cell. Okay. As we inject the titrant into the titrant, what's happening is that these interactions is going to release or absorb some heat, depending if it's an exothermic reaction or endothermic reaction. And therefore, the difference, so the temperature in the cell, in the sample cell and the reference cell is going to be different. And the equipment is going to try to bring these temperatures again to the same temperature. And so the equipment is going to to apply some power to bring this temperature to the reference cell temperature. And we can quantify this energy that the machine is using to make the temperatures again the same. And as we inject more and more the titron into the titron, we're going to have these peaks for every time that there is an injection. It goes back to the same temperature, another injection, it goes back to the same temperature. After, at some point, we have a plateau in here because after all the molecule has been saturated, there's no more interaction. We're not going to have this absorption or release of heat, right? So this is the raw data, but if we make the integral below all these peaks, what we have is the typical isotherm of ITC is this sigmoid in the let's say in the ideal of the cases. Later we can have much more complex interactions that are beyond the scope of this presentation. Today is just an overview. And we can extract some different information from this plot, such as the stoichiometry of the reaction, the affinity, or the enthalpy of the reaction. Let's go a bit in depth into this. So as you know, the stoichiometry indicates the ratio of ligand to macromolecule in the binding. And we can know it because it's the inflection point into the sigmoid curve. Also, the slope of the central part gives us the affinity, or how strong both molecules are bound together. If we know the affinity or this dissociation equilibrium constant, we can know Ka because one is the inverse of the other one, and therefore we can calculate through this equation in here the Gibbs free energy. Remember that for having a spontaneous reaction or binding, the delta Gibbs needs to be negative. This indicates a spontaneous reaction. And if, okay, we also can know the enthalpy of the reaction because the difference between the 0 and the maximum of the plot is going to give us the enthalpy. An enthalpy that contributes to the negative free Gibbs normally indicates the presence of bonds. So electrostatic bond, van der Waals, a hydrophobic bond. And if we know the delta Gibbs and the enthalpy, we can calculate then the entropy, right? And a contribution to the entropy into this binding normally indicates the breaking of conformational restraints, or also hydrophobic interaction, sorry, between both molecules because there is displacement of water. We'll see it more in the examples. Okay. So in here we have like 3 different cases that might be happening, and the case A, B, and C. Okay. And there might be 3 different molecules that are interacting. So 3 pairs of molecules are interacting. In all the cases in blue, we see that itself the binding is happening. We have an spontaneous Gibbs. But in the first case, for example, no, sorry. In the first case, what we have is a great contribution from the enthalpy, which might indicate strong interaction between both molecules. For example, it might be electrostatic. If we know that their molecules have charge, we need to know beforehand a bit our system. In the second case, in the B, we see a positive contribution to the enthalpy. So the binding is happening through other reasons. For example, it may be hydrophobic interaction because we see a lot of the contribution from the entropy, or we can have both of them. Okay. Let's see some examples to help you understand these things, or applications in which we can use ITC, because not only allows us to understand the mechanisms for which they bind, but also to like in this case, it allows us to calculate the CMC of some micelles. You know that micelles form when the surfactants into a system reaches this critical micelle concentration. An experiment to know which is the CMC is one that we can have into the syringe our micelles form, and then we're injecting into the sample cell. As we're injecting it, of course, in the sample cell, the concentration is going to be much lower, and so they're going to be breaking these micelles. And therefore we're having some release of energy that we're putting here. But after we reach some point, the concentration is going to be higher. So we're going to surpass the CMC, and we're going to inject micelles into micelles. So there is no energy because there is no breaking of binds, right? And then we can make the first derivative of this graph in here. and we're going to have this, the first derivative, we're going to be able to calculate the CMC for this system. Okay, so that's what an application that we might have. Other application of ITC, for example, we can discern how the ionic strength of a solution may impact our stoichiometry of our system. For example, in this case, these researchers wanted to see what was the stoichiometry between an anionic surfactant with their arginine-based polymer. So for a concentration 0 so for a low ionic strength without salts in the media, they saw that for every molecule of arginine, there was the binding of one SDS molecule, because this one is charged positive, the other one is charged, so this one is charged negatively, and this one is charged positive, so there is interaction of 1 to 1. But however, as we increase the concentration of those salts in our medium, we see that the stoichiometry is no longer 1 to 1, but instead is, for this case of 200 millimolar of salts, there is a difference in the stoichiometry, it's 0.55. And this could be explained because, of course, if we have a lot of salts into the system, part of this positively charged group can be blocked by the chlorine ions. And also the critical mesyl concentration of surfactants decreases as we increase the ionic strength of the media. So now the molecules are not free to interact with all the arginine. So instead they are forming micelles and they have steric hindrance. Final application of whoa, what happened here? Okay, this glitch, as you can see, I don't know why. It was okay in my computer. Okay, and a bit more complicated or a bit more interesting application of ITC is the evaluation of size dependence proteins onto the corona. We have talked about extensively about the protein corona. So these researchers, what they did was to try to understand how for similar proteins in terms of biochemistry and functional structural properties, how the size of these proteins affected the inclusion into the corona of this silicon nanoparticle. And they studied these interactions of the corona at 2 different pHs. Okay, they made all the plots in here. You can see that for different proteins, they have like different. In here we have an endothermic reaction, and here we have exothermic. Let's go a bit into each of them. Let's take this one, for example, as a reference. So first of all, let's talk about the affinity. The affinity, as I said before, is, or let's say, is how strong some molecules bind to the other ones. So what they saw is that as we increase the size so as the protein increases in size, this constant was higher, implying that there was higher affinity towards our nanoparticle. Also at both at pH 6 and pH 7. However, when we analyze the dependence on pH for both for the affinity, we see that at pH 6 we have higher affinities in comparison with pH 7, and that could be explained because these proteins, these hemoproteins, has a selective point of the histidine moieties at around 6.5. So when we are at pH 6, these histidine moieties are protonated, could interact with the siloxide groups in the surface of the nanoparticle. Okay, that's for the affinities. That's one application that you can have with ITC to check how strong is the binding. Also the stoichiometry. This is pretty straightforward. They saw that for proteins that were much smaller at both pHs, the stoichiometry was much higher. This is quite straightforward. So bigger proteins can attach less of the proteins onto the molecule, onto the nanoparticle. Sorry. And finally, also they wanted to understand the mechanism behind the interaction of these proteins. And in C we have differences so we have here displayed into different colors the different proteins from the smaller to the bigger. In all the cases, we see that the reaction, so the binding, it's happening spontaneously because all the delta Gibbs is negative. But the mechanism behind is different. Okay, so for small proteins, it looked like the binding was driven by interactions, by H-bond or electrostatic or van der Waals. Even if we have some positive or negative contribution from the entropy, probably due to a loss of conformational freedoms in the proteins when they are bound, regardless when they are in comparison when they are free. But however, for the big proteins, there is less interaction, there is less electrostatic or van der Waals interaction between the silicon nanoparticles and the proteins. So it means that probably the binding between the two was due to the hydrophobic interaction, because when a big molecule attaches to the nanoparticle, a lot of water is released. That's probably why they were binding, even if there was some negative contribution from the enthalpy due to the conformational changes that the molecule has to undergo, and they have to break some intraprotein interactions in order to be able to bind with the nanoparticle. So as a summary of ITC, why you may use it. So the good thing is that it measures the heat, and heat is universal. So you can analyze everything without the need of labeling, attaching, or whatever, because the heat is going to be absorbed and released in every interaction. However, since it's universal, in your plot, you're going to have also secondary interactions from other unwanted bindings in your system, or precipitations or whatever. It's going to also release or source of energy. You need to discern which is your event that you want to study and the wanted one. Also, it's in solution. These two goes together. So it's in solution, and you don't need to purify it. You don't need to treat your sample in any way. But it's very time consuming, and as I was saying, the data is quite complex to understand. So you really need to understand a bit your system beforehand. And also it provides you with all the thermodynamic profile. It also gives you the kinetics, but it's not normally used for this. For kinetics, we have the SPR, in which we will talk now. But yes, I was saying it's a bit, the analysis of the data is a bit difficult. So that's it. So now let's talk about surface plasmon resonance. This is a new service that we recently incorporated here into the subservice. As a disclaimer, I have to say that my knowledge about the SPR right now it's a bit limited. Okay, but I try to make you as good as an overview as I can. So let's talk about the plasmons. Plasmons are the collective oscillations of electrons in the interface between the electric materials, or for example a buffer and the metal, when they're excited by light. And so this collective oscillation of electrons generates an electrical field that permeates into the dielectric material. Okay. This is very interesting, because with this phenomena we can characterize these molecular interactions. So let's talk about a bit about the SPR setup. In the SPR setup, we have a light source that produces a beam of polarized light that passes through a prism and is directed into a metal surface. That is normally gold in SPR, although other metals can be. It's normally a gold surface or a gold-coated surface. And this gold surface can be functionalized with some attached molecules that can be bound covalently, for example, to the gold. And then all this light that inflicts into the gold surface is reflected into the detector, and we can read the reflected light. However, as I was telling you before, for some angles of the incident light, there is no reflection, because the energy of the beam is being transformed into the generation of these plasmons. Okay, so for a specific angle, we won't have reflection. We'll have the surface plasmon resonance. However, this surface plasmon resonance has this electric field that permeates into the dielectric media, which in this case can be a flow chamber with molecules flowing. It's highly dependent on the refractive index of this media. Okay, we can change the refractive index by changing the composition of the buffer itself or the temperature. Or another thing that we can do is change the local mass into this dielectric. Like for example, flowing some vesicles, some molecules here that have been attached to the ligands that are bound to the gold. Okay, so in here we have a small video. Okay, so we have our ligand attached to the gold, and as the other molecules bind, the change into the refracted light, in which we don't have reflection, changes. Okay, as they unbound, the angle goes back. So it's a reversible also process. So, but in order to extract the information from the SPR, we need to build what is called a sensogram. Okay, so we have said that we might have our ligands bound, covalently bound, for example, into our gold surface, and for some angle, we're going to have the production of these plasmons, and then this angle, the angle at which the plasma is being formed is proportional, we said, to the mass attached to these ligands. Okay, and through an internal calibration of the equipment, every company that has SPR have their own calibration. This is directly proportional to a magnitude that they call response unit for the SPR. Okay, so at the beginning, when all the ligand is free, we have our sensogram at the baseline. But as we flow molecule that is binding to this ligand, we have the association part in which the angle changes, the mass changes, and as a result, the response unit increases. This is the association part. And then after some time of injection, we're going to reach to a stage where either all the ligand is saturated or we reach an equilibrium where the velocity of attaching to the molecules binding to the ligands is the same as they are releasing. And after we wash with some buffer, we're going to have the dissociation process. Okay, let's see with this sensogram, how can we extract and which information can we extract from the SPR. So for this, let's assume the easiest of the case, which is the Lamuir binding. In this simplification, we don't have mass transport limitations, meaning that the molecules that are attaching to the ligands are already present at the surface of the ligands. Also, there is no heterogeneous binding. There's only one molecule binding with one another. And finally, we inject long enough that we always reach the equilibrium. Okay. As I was telling you, we have in here the baseline. After when we start injecting and the molecules start to bind, we have the association after we reach the equilibrium. And then we have, when we wash with buffer and the molecules are released, we have the dissociation. We can later have or not regeneration in which we clean also the bound molecule, so the ligands, in order to start fresh from another experiments. Okay. The first information that we can obtain from SPR is the binding kinetics. So in this Lamuir simplification, in Lamuir binding simplification, we have two molecules, B and A, that forms a complex. This complex is governed by the association constant Ka, and this is also reversible. And this association is governed by Kd, which are these constants. And for this kind of simplification, Lamuir simplification, the dissociation part is controlled by this equation here. Very, very simple equation. If we solve this, we will obtain a single exponential that fits this part in here, and therefore we can obtain the Kd. Higher Kd means faster dissociation. Of course, you don't need to solve these equations. The equipment does it for you. It will give you directly the Kd. If we know the Kd, we can know the Ka. So the dissociation part depends on both Kd and Ka. But if we first calculate the Kd, we may have the dissociation constant. Again, the same, faster, higher Ka means faster association. If we know the kinetics, we can calculate the affinity, or how strong, as with ITC, how strong both molecules bind. Kd represents the analytic concentration at which half of the ligand binding sites are occupied at equilibrium, and it's pretty straightforward to calculate because it's the Kd over the Ka. So if we know the constant, the kinetics, we can know the affinity. In this case, a higher Kd implies a weaker binding. For example, enzyme substrates or small molecules tend to have weak bindings, and antibodies with antigens tend to have lower KDs, which implies higher, higher affinities. And lastly, we can also use SPR to calculate the concentration of an active molecule that we have into our sample. For that, we need to build a calibration curve so we can record the response units at equilibrium with aliquotas in which we know the concentration. And then we can plot all these response units in this graph, and then we can use a sample in which we don't know the concentration of our molecule we want to study to know the real concentration of our sample. Let's see an example of this very phenomena. For example, in this publication in here, these researchers were trying to, thank you, they were trying to implement a fast and reliable way to calculate the concentration of carcinoembryonic antigen. It's a kind of antigen that is very present in colorectal cancer. They normally, so they traditionally calculate this concentration of these samples with patients with colon cancer with other techniques such as chromatography, ELISA, Western blot. However, these techniques are time consuming, and sometimes they don't allow you to work with non-processed samples. Like, for example, you cannot work in ELISA with pure blood. You need to treat it, or they are not sensitive enough. So what they did was to develop this SPR system in which they were covalently attaching some monoclonal antibodies that have a very high affinity for this antigen. And then they took samples from 14 patients with colorectal cancer, and they calculated through other techniques the concentration of CEA, and then they processed these samples again through SPR, and they saw how many response units were they given. For this part in here, which is they were able to make this calibration curve, and it's very good because from 5 to 10 to 20 nanograms per milliliter is the medically relevant part of this antigen in patients with cancer. And this way, they were able to develop this SPR way to analyze the concentration of this antigen into patients, which you have another techniques, but with SPR, you need pre-concentration of the sample, and you need also really small amounts of sample, and it's very sensitive. Another example, this is more focused in material science, since we are an ICMAB. You can also use SPR to analyze how the density of a monolayer affects its proving capacity of, for example, in this case, oligonucleotides. In this example in here, the scientists wanted to see if, so they attached some DNA probes into a gold surface, okay? And they attached them into different configurations. One which was much dense, and the other was less dense. In here they have the probes were 6 nanometers apart, and in here were 8. And then they wanted to analyze if perfect matches, so sequences with perfect matches of DNA have higher affinity than samples with 1 mismatch or 2 mismatches in the base of these oligonucleotides. For the ones with low density, they saw the expected results. So the perfectly matched sequences were having much higher affinity than the ones with 1 mismatch or 2 mismatches. But with the one in the case in which the probes were really close one to the other, they saw that the steric hindrance provoked by this closeness to the probes was much more important than actually the complementarity of the bases, because they were seeing that they could not reach more than 50% of hybridization, even if the kinetics were a bit faster for the perfectly matched samples. Now, regarding some advantages and limitations, just to finish with SPR, the good thing with SPR is that we have a label-free detection method. We don't need to put fluorescent attachments to our molecules, but it's surface-based, so you need to bind one of the molecules to a gold substrate, which may change the conformation of the molecule. It also provides real-time kinetics. But in here we have seen the laminar binding, which is really ideal, but sometimes you can have problems in the transport from the bulk of the microfluidic chip of this buffer onto the surface. It's very, very, very sensitive, but it's not specific. You can have some unwanted attachments of some molecules to the gold surface itself. And it also works with very low amounts of sample, but you don't waste too much sample, but you need to, sometimes you need to waste gold surfaces because you cannot regenerate always. And it's very versatile. It works with a lot of, you can use a myriad of proteins or peptides or nucleic acids, but you need to see which strategy you need for binding each of these molecules to the gold surface. Okay. Just as a take-home comparison, we have seen both techniques in here. ITC measures heat changes, and the other one, it's an optical method that measures the refractive index. ITC, it's very convenient because you have this molecular solution, you don't need to bind anything, and the other one, even if it's faster, you need to immobilize one of them. The data provided is more complete in ITC because it gives you the whole thermodynamic profile, while SPR only gives you kinetics. And there is no restrictions, and ITC, you can work from micromolar to nanomolar, but SPR is even more sensitive. You can work out to picomolar. And as I was saying, you can quantify thermodynamics, binding energy, stoichiometry, and you can discern the mechanism of interaction with ITC. And with SPR, you can only study the kinetics or calculate concentration. That was all. Just wanted to say that in the self-service here at ICMAB, we have both techniques. So if you want to use one of them, just make sure to contact us in our socials or just send me an email. Thank you very much.
Thank you very much, David. Is there any question in the audience? I will start asking about ITC, because when we are studying molecular interactions in biology, it's usual to have samples that are very expensive or very precious. So I wanted to ask you, which is the volume of sample that you need, for example, for ITC? How much sample do you need?
So that's highly dependent on the equipment itself. Here in the ICMAB, we don't have like the latest technology, let's say. In fact, we have one that it's like 25 years old. So something we need to discuss with direction. But the newer equipments, the ones that are being sold right now, I think it's maximum 300 microliters or so of sample that you need. It's really not that big of a.
And in ITC, how do you choose which one is the titrant and the titrate?
You can interchange them. In principle, the measurements should be the same in most of the cases. So normally you have in the syringe you have less volume, so you choose the one that it's that you have less quantity. But in principle, the interaction should be the same, even if you inject one to the other.
Any question?
Thank you, David. One curiosity about ITC, it is possible to do reversible measurements or competitive measurements, so to add two different...
Reversible measurements, what do you mean exactly? Because
so you study one interaction and then with a competitive ligand, You can exchange the ligand to study competition between one and another and making the interaction reversible. Are there examples about this?
I mean, the fact is that when you inject one into the other, you have this final sample, and it's sometimes not easy to recover it, and you have already the cell filled.
Okay.
So you need to recover it somehow. So,
Okay.
I mean, from the point of view of the technique itself, I think it can be done, but I mean, experimentally,
the design of the experiment. Okay. Yes. Okay.
You could try to recover it and then
I'm
put the
thinking about
antagonist.
this. Okay. I'm thinking about one experiment. So thank you. Thank you very much.
Connecting with this question, maybe you can do that in SPR because you said that you flow some molecules that interact and then you dissociate them. Can you inject some other ligand that competes?
Yes, SPR, you have normally a microfluidic chips, and then you can wash with the buffer, recover your ligand, and then have your ligand again fresh to check with another antagonist or whatever. But normally the data that you want from one is not the same data that you obtain from the other technique. So depends on what you want. One may serve you well and the other may not. But yes, with SPR you can do it.
Is there any other question? This is the last one.
Thank you for the opportunity. David, regarding the ITC, is there any size limitation for the analytes that you are measuring?
Size limitations of the molecule itself, or the?
Because in the examples that you show us, there was a nanoparticle.
Yes.
So which is the size limitation of the nanoparticle?
I mean, for me, as long as it's in okay, this is a bit tricky. As long as so ideally you want to have the molecule solubilized. If it's a suspension, you need to have a really stable suspension. Yes, because if not, you're going to have precipitated into a cell. The cell is stirred all the time.
But over how much time do you need the dispersion to be stable?
I mean, these experiments normally, at least with the newest equipments, are like 30 minutes. But if you're having sedimentation or flocculation over time, then your measurements are going to change, not because of the interaction, but because of the precipitation. And precipitation also produces heat. So ideally, you need to have it completely stable or really, really as stable as you can over time. I mean.
Thank you very much.
David, very nice talk. As far as I understood, in both of them, the reactions that you have, they have to be first order reaction. It means you have two species, and one species, the concentration is constant, and changing the concentration to another one.
You don't need to have first order. I mean, you can have all kind of interactions in here, in both of them. However,
the equation,
the analysis, I mean, the data provided by these two techniques is not really straightforward, okay? So for example, in ITC, in which I have more experience, you normally have much more complicated interactions between between molecules, and then you have plots that you really need to understand. That's why you normally do a lot of plants, like one protein, one of the molecules into water, water into the molecule. These are experiments to try to eliminate what is not interesting for you. Right. If you have a very complex interaction, then you will need probably another technique to complement, to understand what's happening inside.
Also in SPR, just a curiosity, is it possible to study degradation of one process? It means you have something attached from the beginning, and you want to study the releasing of this rate of releasing these species. Is it possible to?
Is it possible? Because the refractive index is going to change over time, right? Because so this measures the angle changes because the refractive index changes if molecules get attached, but if molecules go out or change, you're also having a change in the refractive index.
Also temperature studies is possible in SPR, also lower.
Yeah,
okay, thank you so much.
Let's thank David. Thank you very much. And I will move to the next speaker.